and thanks for joining the 2DCC webinar today. Today's speaker is Dr. Tetyana Ignatova from the University of North Carolina at Greensboro. And Tetyana, you are good to get started.
Good afternoon, everyone. Yeah, it's already an afternoon. So today the topic of my presentation will be molybdenum disulfide, two-dimensional molybdenum disulfide, lattice rejuvenation through photon-induced sulfur vacancy passivation. And first I would like to introduce myself and my group, what we are doing. At nanoscience department, my group works with low-dimensional materials with graphene, transitional metal chalcogenide, their heterostructures, and not only with two-dimensional, we still work with one-dimensional materials, with nanoparticles. With two-dimensional materials, we do defect doping engineering. We work to probe chiral properties of this material. We study charge transfer, modulation of optical conductivity in collaboration with Dr. Rodkin from Penn State. We also try to make devices for stress and strain sensing and applications which we are trying to explore is optical biosensing, bioimaging, and multiple other techniques. And of course, what should I do without my graduate students? You can see them here, and we some of them already graduated, and couple new students joined my lab this semester. Okay, today's talk will be about two-dimensional materials, and we know that there are a big family of these materials. We have transitional metal chalcogenides, which could be semiconductive or could be semi-metallic. There is, of course, graphene. And my topic today will be about molybdenum disulfide, very famous one. And here you can see the Raman signature of this material and photoluminescence, this semiconducting material. And here is my outline. After a very short introduction, I will talk a little bit about optical non-uniformities of MoS2. I will discuss with you defect and doping engineering. How can I change it? 1 second. Then we talk about photo-induced changes and our idea of MoS2 lattice rejuvenation through photodoping. Then I will finalize with conclusions. Can you hear me?
Yes.
By some
reason,
we can hear. My slides stopped. Come to the next one. Resume. Still stuck. Oops. Oh, perfect. So molybdenum disulfide belongs to TMDC family, has and extensively explored for applications and for optoelectronic devices. MoS2 has a direct band gap, and because of it, it became one of the ideal candidates for light-emitting devices. And here you can see just the cartoon for MoS2. Unit cell contains 1 molybdenum atom, 2 sulfur atoms, and thickness of this monolayer is about 1.7 millimeters. On the right side, you can see Raman signature of MoS2, and it's typically in plane, out of plane, 2 peaks of in plane, out of plane peak. And this information actually very useful for us to identify how many layers of molybdenum disulfide we have, and applying more extensive analysis to the Raman data, we can extract information about strain and doping in these materials. As grown material, MoS2, typically N-type semiconductor. And so far, people connected with intrinsic sulfur vacancies. And this is drastically influence photoluminescence properties of MoS2. You can see here photoluminescence graph, which contains two major exciton peaks, exciton A and exciton B, as well as trion peak, and trion is negatively charged exciton. So position, intensity, and width of photoluminescence peak is drastically dependent on local molybdenum disulfide doping level. What I wanted to tell here. Yeah, let's go further to exciton and trions. I think many of you probably know that strong Coulomb interaction between of exciton and charge carriers forming these trion peaks. And this trion consists of 2 electrons and 1 hole. So trions act as a recombination pathway observed in photoluminescence spectroscopy. Why I cannot, and this bothers me, I cannot move to next slide by some reason without. Okay, here we are. You can see here 2 spectra. One will be molybdenum disulfide as prepared, and second one molybdenum disulfide after we doped this material with P-type organic molecule. And you can see that and it was done immediately after growth. And photoluminescence peak had significantly changed. Not only changes in intensity, but also it is shift in photoluminescence maximum. So you can see that just by simply chemical doping, we can change photoluminescence and manipulate band gap of molybdenum disulfide. To probe how much we dope material, we applied Kelvin probe KPFM techniques, and just comparing results with calibrating these results with some reference material, we can calculate how much we move doping level and what is happening upon applying this p-dopant on the top of as pristine as grown molybdenum disulfide flake. Okay, yeah. Optical non-uniformities of molybdenum disulfide exist immediately after you take material from the furnace after growth. And it is probably because of some impurities, atomic defects, because of immediate absorbates, because introduced strain from substrate, number of layers and appearance of second, third layer formation, because of grain boundaries, wrinkles, tears. So many different reasons can cause non-uniformities of as-grown two-dimensional MoS2. And here you can see photoluminescence map for sample. And this sample was actually covered on the top with graphene, which it doesn't matter for now, but you can see that the photoluminescence profile is not flat at all. Such type of response we could analyze by applying Photon and extracting information about intensity, position, and width for B exciton, A exciton, and trion, and then make some interesting conclusions. Okay. Here is our data when we extracted this information, and you can see here that this is the difference in intensities. This is information for exciton and tryon, and difference shift for the widths. So, and by plotting a correlation between information related to exciton A and tryon, we can get some correlation. This data is not correlated well. However, we can see that there is anti-correlation. Enhancement of exciton intensity will be combined with decreasing of tryon intensity, which could be used in future could be used in engineering of optical properties of molybdenum disulfide by applying certain procedures, certain engineering procedures. Optical, of course, we know that optical characterization is always limited by diffraction. And to enhance resolution, we can correlate this data with tip scanning tip techniques like AFM or SNOM, and then we can correlate and kind of create higher resolution for this optical characterization. And we can, for example, at this slide, we calculated charge transfer in van der Waals materials, graphene MoS2, by applying KPFM and correlating KPFM and SNOM. And this type of correlation procedure allow us to calculate exactly the amount of charge transfer in this heterostructure. Okay. And then combining all possible techniques, it is possible to create multidimensional images of two-dimensional materials. For this particular slide, you see that it is MoS2 covered with graphene, but it is possible to do for any two-dimensional materials combination or just for one single nanomaterial. Okay, how to quantify molybdenum disulfide defects? To look at this problem, we brought molybdenum disulfide material into helium ion microscope, and we controllably introduced defects. And then we applied, you can see here that results for scanning transmission electron microscopy. There are certain amount of defects which were existed from the beginning, but then we introduced more. And then we applied quantification by combining, correlating multiple characterization procedures, just analysis of STM data and Raman. You can see here that how defects were created, and these defects, some of them, each time we quantified these defects and we identify how many atoms are missing, and we plot inter-defect distance depending on helium ion dosage. We also brought the same sample to Raman instrument, and we took Raman data. And Raman data showed us significant changes in two Raman peaks, main Raman peaks with increasing of helium ion dosage, but as well we found the appearance of longitudinal acoustical mode at 227, which is attributed in literature with scattering from M point, defect-induced scattering from M point. And then we combine together Raman characterization and our analysis of our STM data, and we extracted and calculated the correlation constant, which will be which appears to be in linear correlation with Raman data. Okay, with this knowledge, we can apply with this knowledge, we can do band gap modulation, MoS2 band gap modulation, when we in purpose introduce defects and combine it with chemical doping. So the same sample, but what we did, we applied additionally on the top of chemical doping. Let's look here how characteristics of this sample will change. For the pristine sample, which here represented as a black curve, and on the left upper corner, you can see photoluminescence spectra. When we add more and more helium ions to the sample, you can see that the photoluminescence start to be quenched. So from black, we go to green, to red, to blue, to purple, to the green, which is, I can say, disappearance of material. At the same time, the Raman peaks will show us this shift of the Raman peak, which could be also analyzed for future purposes. The same sample was then defected and doped with p-dopant, and you can see that it was started with black curve, the same as here, and then instead of quenching of photoluminescence, we observe enhancement of photoluminescence as well as shift. Enhancement, and then after a certain value, it started to be quenched the same way as you can see for photoluminescence on the just defected sample. Results of this method is presented on the right panel. You can see that for multiple doses of helium ion irradiation, we have enhancement or quenching of photoluminescence. Okay, we can quantify the disturbance we introduce by applying this mass action model, where we calculate concentration of excitons, trions, and excess of electrons, and again correlated with photoluminescence intensity. So it was done. We calculated, we found that the applied doses caused us a reduction of electron concentration in excess, and it was in a perfect agreement with results we can see from the panel on the left. Here is additional calculations. Okay, let's go to the next slide. The topic of my presentation is molybdenum disulfide rejuvenation. Initial observation of photo-induced changes are presented on this slide. It happened a few years ago when we found that by doing characterization, we already introduced changes. These ugly pictures were done at old Raman microscope when student just took out sample, freshly grown, sample was bad, and he started to do mapping, Raman mapping, and already see that scanning across will cause significant changes in appearance, just optical appearance. That is optical characterization, nothing special. We also see in correlated Raman changes, we definitely see that at certain point, sample became more clean, then sample did not change much, but after applying more and more power, samples start to disappear. So we definitely saw some cleaning, then some changes, and sample became better and better, and then samples start to disappear. It was photo etching. Of course, there are a lot of publications, and right now already existed, and we checked what information is already available. What we found was that laser can induce thinning of the material and cleaning of two-dimensional material, which we definitely see. What we also found that effects of photo-induced changes were related to the introduce of trapped defect from silicon oxide substrate. And there is also information that sapphire does not introduce doping because it has no charge impurities on the surface. We also found interesting paper where photodoping was combined with introduction of sulfur vapor, and people saw significant changes in photoluminescence, and they attributed these changes with definitely with rejuvenation of sulfur defects in presence of sulfur vapor. You can see here the results from the paper. Okay, then what we did? We tried to examine sample which was freshly grown on sapphire sample provided by Penn State. Thank you for you guys. And this particular sample, and then we applied the advanced fitting analysis, and we looked how MoS2 on sapphire will respond to introduce laser irradiation. You can see here photoluminescence spectra of such sample which we fit with B exciton peak, A exciton peak, and trion. Of course, even from the beginning, as grown sample has a little bit of trion influence, which is fine. What we did later on, we illuminated this sample with power which will not cause overheating, will not cause etching, and we traced the optical signature over a long period of time. You can see here on the left side appearance the maximum center location of photoluminescence spectra over a certain period of time. You can see that there is enhancement, significant enhancement, and this sample was not aged, it was freshly grown sample. Enhancement, but with 3 slopes. We did not present here, but after this period of time, we started to see the decrease of photoluminescence, slight decrease of photoluminescence. And on the right side, you can see how spectral lines look like. On the beginning, so after illumination on middle point somewhere here, you see that intensity of photoluminescence jumped up almost twice, and we see significant shift of the maximum of photoluminescence line. That gave us an idea that it is not the reason for, okay, let's go to the next material, which of course on silicon oxide, transparent material, always showed us significant change in photoluminescence. You can see it here, this sample is not provided by Penn State. This sample was grown here at Joint School of Nanoscience, Nanoengineering. But the same situation happened with material which we transferred from sapphire to silicon oxide. The difference was that, of course, during transfer, we have much more defects. So that material did not sustain so long as on sapphire, and we see the decrease of photoluminescence intensity much faster. Yeah. What we did statistical analysis of photoluminescence maps, and what we see, we definitely see data clustering, and we see intensity anti-correlation between trion and A exciton. And this allow us to make conclusion that it is a conversion of trion to exciton upon laser illumination, upon introduction of certain power. And even we can extract certain slopes because we illuminated during over a certain period of time and extracted the information about optical response of material. Of course, we brought this sample and did Raman analysis, and we looked how AFM profile topography will change over for the sample which were illuminated with laser, irradiated with laser. This particular picture shows that definitely AFM before and after became much more smooth. And this is still, we are still thinking what is happening because we are not removing contaminants from the surface. MoS2 became absolutely flat, and we don't know why. We found that it is significant reduction of strain, but when we increased power here, you can see here that reduction of strain increased even more. For this particular picture, there is, I don't know, probably some crack coming through the sample. This is the explanation of this line. But we can see also and calculate how much doping we introduce just by irradiating sample with certain laser power. And I think I'm very short because I can now jump to my conclusions, and that we see that sulfur defects modulate molybdenum disulfide band structure, and we see that defects exist in as grown MoS2 and transferred material. And we see that laser treatment induces lattice healing and enhanced photoluminescence response. And with this, I would like to thank you, my two graduate students who, Anthony has started this research, and Anna is right now polishing, finalizing this project. We still need a lot of the finalized experiments and make final conclusions. I thank you, collaborators, to the RSVP program for help for my students, as well as collaborators, Dr. Arava Muthan Group, Dr. Rodkin Group, and my collaborators from DevCom Soldier Center. Thank you very much.
Thank you, Tetyana. Does anyone have any questions?
I can have a question. So when you did any of the measurements for Raman or PL, were you doing those in vacuum?
No, ambient conditions.
That's a problem.
I know.
There's papers already out there that talk about how just any ambient, like water, I mean, even your breath, like your coffee breath that you had in the morning, will dope the system. And there are laser powers much lower than even the powers you were showing, 10 milliwatts for the power, which is actually
no. 10 milliwatts, it's too much. Major experiments were done below 1 milliwatt.
Yeah, even at like microwatt, at 1 microwatt, you can actually already see desorption of species off the surface of MoS2 that will change the luminescence. So, I mean, one thing that would be beneficial is, you know, to really identify contributing factors to the change in the PL. I mean, it's no surprise that you're changing the PL with the laser. But you want to be able to attribute that to a specific either defect or whether it's desorption of a gas. If you can do it in vacuum, that's the most ideal. Or if you do it in an environmental chamber where you're purging it with like very dry air or
nitrogen,
you know,
right?
Argon would be preferable. That would maybe help to isolate what is actually happening as a function of laser irradiation. But yeah, certainly, I mean, I agree, you can change this material.
I totally agree. But what brought us to our conclusions that we always see the same shift. I showed only one correlation plot, but we always have very, very similar. It really depends on number of defects or if sample is fresh or aged. Then it will be, the ranges will be differently splitted. But yeah, I totally agree. We definitely look for vacuum Raman analysis in order to make conclusions. Yeah, thank you. Thank you for this comment. And we also traced the anti-Stokes peak just to see if we are not hitting sample. What we wanted to do, not hit our sample. And yeah, this is what we also always checked.
Yeah.
Go ahead.
Yeah, so just a question on your third conclusion point there. You say lattice healing. Could you talk a little bit more about what you think might be going on there structurally with this healing of the lattice? What defects do you think you're healing, and maybe how they're healing?
For the sample which is grown on the top of silicon oxide, CVD grown samples at our institution, we know for sure that we have excess of sulfur. This is for sure. And I think for this particular picture is just optics. But I think that introduced laser power gives enough energy to give enough energy for sulfur to be incorporated into defected spots. We are doing what we are doing simultaneously cleaning surface, but also excess of sulfur comes to the defected spots, and this is why we think it is lattice healing. We are still trying to provide more evidence for this. Yep.
Interesting. And just to follow up on that, and you believe this to be like a photocatalyzed effect and not just a localized heating effect? You'd mentioned trying to minimize sample heating.
Photocatalyzed effect, yes.
Okay, so if you thermally cycle this, you wouldn't expect to see the same amount of healing that you're seeing with the photoirradiation?
For this, we have to do it again in argon atmosphere, right? And we definitely see enhancement of photoluminescence properties after thermal treatment in argon atmosphere. This is true, yes.
Interesting, very interesting.
Tetyana, there's two questions in the chat. What is the laser wavelength used? And then Slava had a question.
Okay, we used 532 and we used 488.
And then Slava's question is the optical image IBN conclusions for transferred or as grown material?
This optical, this is the grown material, not transferred.
So if I may follow up, so do you have an idea what are these particulates that are being cleaned here? Are they coming from the growth itself or some post-processing? The small particles that disappear upon photo cleaning. So that's the growth.
Growth. Growth for sure. Yeah. What is interesting, why we are thinking about healing after this enhanced optical response is not temporal. If a sample was treated, how can I say, perfectly to the maximum of enhancement, it will hold this optical response over days until just normal agent factors will influence significant suppressive optical response. Yeah.
And I did get it right that it happens pretty much equally for the silicon oxide substrate, which has photo traps, and for sapphire, which does not have any photo traps.
Yeah. Yes.
Okay, thank you so much, Tetyana. We appreciate it.
Mhm.
Thanks, everyone.
Thank you. Mhm.